today's talk is going to be by someone who I think is well positioned and well suited to carry forward on this concept of potential, of promises, what stem cells, what stem cell research can do for us. Professor David Schaffer, who I see on the top row of my screen, I don't know where he is on yours, is actually an ideal candidate for do this for a number of reasons. In particular, his own research applies engineering principles to the study of stem cell and gene therapy related to neuroregeneration. That's the nervous system. So it's one of the very exciting areas that stem cells are opening up. And he's well qualified for this because he's a professor of chemical and biomolecular engineering, of bioengineering, and at neuroscience here at Berkeley. And he's also served as the director of the Berkeley Stem Cell Center. And on July 1st of this month, just a couple of weeks ago, there was an announcement that he was appointed to be the new director of the Berkeley Institute for Quantitative Biosciences, which is called QB3. And if you don't know about that, I encourage you to go online and look it up. This is an initiative that started in 2000, and it was related to the renovation of Stanley Hall. And it epitomizes the idea of multidisciplinary research, because it brings together people from different departments at Cal, as well as from UCSF, UC Santa Cruz. It came from a big grant, government money, to get this interdisciplinary thing started. Dr. Schaffer actually joined the faculty here, I believe, in about 1999. He started his undergrad work inauspiciously at the small university down south. I think it's called Stanford. And then he went on to get his PhD at MIT before coming to Berkeley. So he's been very much involved in the center of the exciting new field. And I would like then to welcome very much Dr. David Schaffer. David, take over.
Thanks so much. I really appreciate the introduction, as well as the chance to share some thoughts, as well as a little bit of research with you. I'm going to switch over to sharing a screen real quick. Okay, great. And so I have the format of my talk is that about the first half of it is introduction, just conceptual discussion about the potential within the field. And then the second half is a little bit more diving into research, and specifically talking about how to produce these cells to be able to treat large numbers of patients who had the potential to benefit from cell therapies. And so I'm going to have half the talk be pretty conceptual, and the second half a little bit more scientific. And if there are any questions along the way, as I mentioned, please feel free to just pipe in through chat, and I'm happy to pause for a second just to make sure everybody's on the same page. Okay, so the goal is to talk about, I know the concept of human pluripotent stem cells, or stem cells in general, were introduced last week. And today I want to talk about the potential to apply these cells towards treating human disease. And this is really the motivation right here, which we're all familiar with, that regardless of which particular tissue you look at within the body, there are a large number, unfortunately, of diseases that represent unmet medical needs, diseases for which there are treatments in some cases treatments that ameliorate symptoms, others that slow the progression of disease, but in most cases, there's not actually cure that's available for these conditions. And so these obviously rob people of their quality of life. And in addition, they drive healthcare costs. And so this is a projection, both currently as well as into the future, of the cost of healthcare that's spent, the amount of money that's invested in this country in healthcare. And so back in the 1960s, it was single digit, around 5% of our gross domestic product was spent on healthcare. That's risen to double digit. The most recent years that we spent around 18%. And this number is projected to continue. And these long-term chronic conditions, in many cases, drive the shape of this curve, drive the cost of healthcare, because these progress on a time scale of years to decades. And as I mentioned, don't have a cure, but have treatments, in many cases, ones that are rather expensive. So if we take a step back and try to look for potential ways in which we could treat or even cure these conditions, I want to roll it all the way back to what's called the central dogma of biology, which is kind of taught the first lecture that anybody takes a biology course. And so within this dogma, we look at DNA, and DNA, of course, is kind of the repository of information inside of a cell, inside the body. It's the hard drive. And this has information in the form of genes, and that information gets transcribed into RNA, which then ultimately gets translated into proteins. And proteins are, of course, the functional side of biomolecules inside cells and inside the body. So RNA and proteins together are responsible for the function of our cells, and the information that encodes them is present within the DNA. So malfunctions in these three levels are what drive many of those diseases. And so if we're trying to develop therapies to treat these diseases, we begin to look at each one of these levels of biological information as a drug target. So the vast majority of pharmaceuticals and biotech products that are on the market today operate at the level of DNA. So we know small molecules, things like ibuprofen or aspirin, and these typically are ingested, distribute throughout the body, find their targets, and typically bind to an active site within an enzyme, within a protein, and change or inhibit the activity of that protein. And that has some desirable outcome in terms of improving the symptoms of the conditions associated with the disease. And there's another class of molecules, monoclonal antibodies, that have arisen as very important within the biotech industry. This is the reason that Genentech is as successful a company as it is. They rose really with monoclonal antibodies and pioneered that class of therapeutic. And these are similar in some ways to small molecules. They typically float around on the outside of cells and find a target and bind to it, and most often inhibit, block the activity of that target. So these really have transformed medicine over the past century, small molecules and protein therapies. But they do have some limitations. And one of those significant limitations is that they have a finite half-time or residence time inside the body. Small molecules typically last on the order of minutes to hours, and that's why we have to pop an aspirin every 6 hours if we have a prolonged headache, because the aspirin only lasts a few hours and we need to keep redosing. So there is opportunity for moving to the other two levels of biological information and drugging them, using them as drug targets. So if we move up one level, there are interventions that operate at the level of RNA. There's something called RNAi, something called antisense, which typically block the function of messenger RNAs. There's another one here where you can actually deliver a coding messenger RNA itself. So RNA itself could be potentially a drug, and this is the business behind Moderna, which you may have heard of recently since they're involved in vaccine development for COVID. But today I want to talk about that other level of biological information, DNA. And this is really where my entire career has revolved around, where if you deliver a piece of DNA as a medicine, that would be called a gene therapy. If you change the sequence of existing DNA inside of our cells, that's called genome editing. And if you add brand new cells along with their genomes to the body, that's called a cell therapy, or for example, a stem cell therapy. So the advantage of working at the level of DNA relative to RNA or proteins is that DNA, unlike all these others, is a permanent part of an organ or a tissue. So if you add a therapeutic gene or add a brand new genome, a new cell to a tissue, that has the potential to persist for many years or even decades. And so you can begin to think about the idea of one and done, single administration, long-term therapeutic benefit. And that really addresses some of the shortcomings associated with small molecule and protein therapies. And it really has the potential to be a strong match for the long-term chronic degenerative nature of those conditions I mentioned to you earlier, because you have the possibility of a single treatment, a single intervention, resulting in a long-term therapeutic outcome that could both improve the quality of life of patients and possibly, since this is a single intervention, a single treatment, could even conceivably reduce the cost of healthcare. So any questions here so far? Okay. So that brings me to cell therapy and specifically stem cell therapy. And I want to talk about, first of all, what a stem cell is. So we all started out as a fertilized egg, a zygote. And once that single cell began to, start on the process of turning into embryo, it divides in a 2 to the nth fashion, 2, 4, 8, 16, 32, 64. And once it reaches a ball of 64 cells, this early stage of an embryo, it's called a blastocyst. And at this stage, cells begin to, initially they're all identical to each other, but at this stage, they begin to acquire identity. They begin to start specializing. And there is the so-called extraembryonic cells or tissue. And so shown here in orange, these are the cells will actually implant into the uterus and start forming the placenta. And then there's the so-called inner cell mass, the cells shown here in blue. And these are the cells that will continue to divide and start specializing and will eventually give rise to an organism. So this is obviously has been done billions and billions of times throughout human history. And there's obviously a very special set of cases due to in vitro fertilization where this is actually done outside the body instead of inside. So starting on the order of around 35 years ago, people began to recognize that in some cases, couples had a really difficult time conceiving. And in vitro fertilization was developed as a technology where a number of eggs, typically on the order of 10 or fewer, were harvested from the mother and cultured in vitro, cultured in a cell culture dish. And then sperm from the father was added to them in order to fertilize them. So then you end up with a plate, a cell culture dish full of on the order of a half dozen to a dozen embryos. So several of these are implanted then into the mother with the hope that they'll end up, forming a child. And so because, 2 to 3 are implanted, this is why there's a higher incidence of having, doublets or triplets from mothers of IVF. But what that means then is that there are a large number of embryos that aren't implanted, and these are quite simply frozen down. And they can be thawed later on just in case the first in vitro fertilization trial didn't work. But in many situations, there are a lot of leftover embryos. So IVF has been used in this country for between 35 and 40 years, which means that if you add them up, there are around 400,000 frozen embryos on liquid nitrogen stored in fertility clinics across the entire country. And in many cases, parents are doing this in their 20s and their 30s. And if you do the math, 30s plus 35 years means that the oldest parents associated with IVF are now in their 60s. And what that means is that they're done having kids. They're not going to use those embryos. So back in the 1990s, investigators began to come up with the idea of why not take some of these embryos, which would otherwise be destroyed since the parents don't need them anymore, and try to isolate stem cells out of them. So back in 1998, an investigator at the University of Wisconsin named Jamie Thompson became the first person to take this inner cell mass, the small cluster of cells from a blastocyst stage embryo, and then put it onto a cell culture dish and figure out the right set of nutrients, the right broth, to keep those cells alive. And that became the first example of a human embryonic stem cell. So this cell can divide indefinitely, spin around this loop, and make many more copies of itself, or it can begin to specialize. And it can specialize, for example, into a tissue-resident stem cell, such as a stem cell of the nervous system, a stem cell of the blood system, a stem cell of the skin. And then these more specialized stem cells, like, for example, a neural stem cell, will continue to divide and make more copies of themselves. And then they can differentiate into non-dividing functional cells, which are the cells that make up the tissues, the majority of tissues inside of our body. So just carrying forward that example of a neural stem cell, this has the ability to make glia like astrocytes and neurons, the functional cell types of the nervous system. So as I'll mention in a second in more detail as well, it turns out that stem cells don't just exist in embryos. They don't just exist in the process of turning a 64 stage ball of cell into an adult organism. We actually also have pockets or niches of stem cells throughout our adult bodies. So our blood system is continuously turning over, and what makes that possible is blood stem cells. Our skin is continuously shedding, so we have skin stem cells that will continue to divide and differentiate. And all these stem cells, regardless of where they come from, share two defining properties, though. They can spin around these loops, meaning that they can make more copies of themselves, which is a process known as self-renewal, to maintain their pool. And they can move from left to right within the slide. They can differentiate or specialize into the functional cell types that make up the adult body. So those are the two defining properties that all stem cells share. So I mentioned to you earlier that one interesting population of stem cells is an embryonic stem cell. And this is what is one type of what is a so-called pluripotent stem cell. So a pluripotent stem cell simply means that this cell has the potential to turn into every single cell type within the adult body. And once it turns into a more kind of restricted tissue-specific stem cell, that would be called a multipotent stem cell, meaning it can turn into multiple cell types, but not all cell types within the adult body. So a pluripotent stem cell is something special. It's something very powerful because you could use that cell to generate many adult cell types. And there are a couple of classes of these. One is the one I just mentioned to you, where you take this ball of cells, this blastocyst stage embryo, and you isolate the inner cell mass, plop it onto a dish, and expand it, and that generates a human embryonic stem cell line. And there's another way to generate a pluripotent stem cell, a human pluripotent stem cell. And this was actually discovered originally back in 2005 for mouse cells, 2006 for human cells, and very rapidly, this was actually awarded the Nobel Prize in physiology or medicine back in 2012. And this was for the discovery of what are called induced pluripotent stem cells. And in this case, you take an adult cell, this, typically a skin cell, just because it's really easy to harvest those from our skin. And then you take these adult cells and you introduce into them certain genes that, when overproduced, will actually kind of turn back the clock within this adult cell, and will effectively reboot the genome and make this adult cell into a brand new pluripotent stem cell, into something that's kind of tricked into thinking it's an embryonic-like stem cell. And so functionally speaking, these induced pluripotent stem cells and these embryonic stem cells are essentially identical. They have the ability to divide, and or they're both pluripotent. They have the ability to turn into every single cell type within the adult body. So any questions so far here?
We do have one in the box. I don't know, Paul, if you want to grab that or if you would like me to read it. Why don't I just go ahead and read it for this time?
Well, I can get that.
Do you have it? Okay.
Yeah, the issue is perhaps going to be addressed shortly, but can stem cell treatment reverse the progression of a disease, or will it just stop the progression?
Yeah, so it has the potential to do both. I'll give you an example. So Parkinson's disease, and we do work on Parkinson's disease. By the time that somebody ends up getting diagnosed with Parkinson's, Parkinson's, it turns out, is due to a loss of a particular population of neurons called midbrain dopaminergic neurons. By the time that somebody is diagnosed with Parkinson's, they've lost typically between 50 to 80% of those cells. And you can function with 50 to 80%. There are Parkinsonian symptoms that are very unfortunate, but if you could stop it there, it would be tolerable for patients who are at early stage Parkinson's. But then there's a progressive unfortunate decline where all the rest of the cells are lost, and then ultimately people end up losing their lives to Parkinson's in some cases. So in the case of stem cells, the goal is to implant brand new dopaminergic neurons that are derived from an embryonic stem cell or an induced pluripotent stem cell, and kind of repair that neurotransmitter signaling system within the brain. And if you can replace the cells that are lost, that would have otherwise have died, you have the potential to reverse the pathology, reverse the symptoms of the disease. So that's why there's so much hope. And with these types of therapies, you can rebuild the tissue. You're adding back cells that had been lost due to the disease.
Is there a risk you'll add too many?
So there are, that's the dream. There are risks associated with this. One of the risks is that if you make a mistake and you add a cell that's dividing still, all of our work is focused on adding cells that are already, quote unquote, terminally differentiated. They're no longer dividing. They're already a specialized cell type. But if you added a cell that's kind of too early, it could potentially continue to divide. And that's viewed as a risk, because then you can't control the cell number. So that's something that we just have to be sure when we enter into human clinical trials that we don't do.
Thank you.
Any other questions? Nope. I'll just periodically take a look up here to see if there are any chat questions that came along. All right, so here's a figure that shows all the different cell types that you could potentially turn a pluripotent stem cell into. So I'm going to kind of go around this loop and kind of paint a picture to you for the potential therapies that could be achieved if you could do this really well. So beta cells, this is the cell type that's lost in type 1 diabetes. So these are the insulin secreting cells within the body. So if you could generate these in sufficient quantity, you can implant them, and this could serve, effectively as a pancreatic transplant, where you could actually reconstitute the insulin secretion system inside the body and begin to treat somebody who has type 1 diabetes. Hepatocytes, this is the major functional cell type within the liver. So if somebody's suffering from liver failure or cirrhosis, this is a potential therapy. Cardiomyocytes. So there are 5 million plus people in the United States who have congestive heart failure and progressive degeneration of heart tissue. So a graft of cardiomyocytes, of heart muscle cells, could potentially serve as a therapy to be able to treat that. Erythropoietic cells. So there are a large number of people suffering from anemia. These are the red blood cell precursors that could potentially serve as a way to reconstitute the oxygen carrying system inside the body. Retinal pigment epithelium, so macular degeneration, age-associated macular degeneration, which results in blindness in over 1 million people in the United States. It's the most common cause of blindness in the US. Some forms of this are caused by a breakdown in a barrier between blood vessels in the eye and the retina. And so this barrier is the so-called retinal pigment epithelium, and blood vessels break through this barrier, start growing into the retina, and that's what causes a lot of the problems associated with a major type of macular degeneration. So if you can implant a new sheet of retinal pigment epithelial cells, you could re-establish this barrier, and that's being used in human clinical trials as a treatment for macular degeneration. Photoreceptors, there are many diseases that kill off photoreceptor neurons, the cell type in the eye that senses photons and are responsible for vision. So retinitis pigmentosa is probably the best known of these genetically inherited conditions. So if you could grow new photoreceptors in a bioreactor and implant them into the eye, you could potentially repair vision from people whose photoreceptors are dead due to retinitis pigmentosa. I'll be talking about oligodendrocytes in a little bit, as well as other types of neurons, because this is the area in which my own lab focuses. Any questions? Okay, so that's the hope. And there's a challenge associated with that, though, which is that, this process by which our body forms all those cell types, inside the adult body is really complicated. We all started out as a single cell, and we ended up as on the order of 10 to the 14th cells, on the order of 100 trillion cells. And we're complicated anatomically, right? We have lots of different cell types. They're organized geometrically in very complicated ways. And it's really hard to, pluck a stem cell out of the body, stick it onto a cell culture dish, and know exactly how to teach it to turn it into the right cell type. So as I mentioned to you, stem cells have two properties. They can self-renew and they can differentiate. And so if we want to take this stem cell and tap into its potential to the fullest to be able to create these various cell types, we need to know how to control these arrows. We need to know how to control the expansion of that stem cell so we can turn a small number into a very large number. And then we need to control or understand how to guide these cells into the right fate so that we can progressively cause the cell to specialize from kind of a blank slate, immature stem cell into, for example, a particular type of neuron that we might need in order to treat Parkinson's disease. So the rest of the talk is going to be focused on kind of understanding how to control these arrows, and especially understanding how to scale up the control of those arrows so that we can generate sufficient numbers to treat large patient populations, each of which would need a large number of cells in a transplantation. So these arrows are controlled by information from the environment surrounding a stem cell. So these cells, whether they're inside an embryo or blood stem cells or skin stem cells inside of our adult bodies, inside of every one of our bodies, they have to be controlled by their environment. If, for example, I start running a lot at high altitude, my body is going to become starved of oxygen. And if I do that long enough, my body's going to start sending some signals to my blood stem cells saying, make more red blood cells. You know, I need a higher oxygen carrying capacity. Or other examples include, you know, skin. If you end up abrading your skin repeatedly, it's going to stimulate the skin stem cells to produce more cells, to produce more skin. So our body has the ability to kind of on demand figure out environmental needs and send chemical signals to these cells to tell them what to do so they can supply the needs of a developing organism or of an adult tissue. So in other words, the tissues, the body is continuously sending signals to these stem cells to tell them what to do. So these signals take a number of different forms. They can be small molecule signals, they can be protein signals, they can be solid phase signals. And this ends up being a really complicated problem because it turns out that these signals are not simply presented to a cell in binary fashion. It's not like the signal is there or it isn't. There can be variations in the presentation of that signal as a function of dosage and time that can represent or lead to a very complicated set of cues that that stem cell has to listen to. So for example, let's say that the cell is listening to simultaneously a half dozen different signals, some of which are actually varying temporally in some really complicated pattern. We need to understand how this code works. We need to understand how to feed these signals to the cell as a function of time to progressively guide it from, you know, this blank slate in the middle so that these cells will kind of travel along the right set of arrows and eventually end up being the cell type that we need to be able to treat a disease. So that's a complicated set of problems within the stem cell field to understand how these various arrows, which represent fate decisions, as this blank slate early stage stem cell is progressively restricting itself and committing itself towards particular fates. We'd like to understand what controls these arrows, and then we need to scale up the control of these arrows so that we can make enough cells to be able to treat human disease, especially ones that involve large numbers of patients. Okay. I see there are questions. I'm going to pop open the chat box really quick. When you add genes to force a stem cell back to a pluripotent stage, can this alter the proteins formed either in amino acid sequence or folding properties and possibly then be regarded as foreign to immune system? So what in pluripotency reprogramming, what you'd like to do, so here you're taking this adult cell and you're transiently overexpressing or introducing these genes, and then that reboots the cell into an embryonic-like stage. So these are all naturally genes that are present inside of our body, inside of essentially every single cell inside the body. And what you just want to do is to transiently, for a short period of time, overproduce the proteins encoded within these genes. And then these proteins will actually kind of reboot gene expression patterns inside the cell and revert that cell to an embryonic-like stage. So these proteins are naturally present inside of our bodies, inside of cells. You just kind of need to transiently turn them on so that they're present at a high amount for a short period of time, for the period of a few days to a couple of weeks, so that you can do the rebooting. So ideally, the way that this rebooting is done is that you transiently introduce extra copies of these genes, of these coding sequences, the cells reprogram, and then those extra copies go away. So there are a variety of ways to do that, but the goal is to do this rebooting in a transient way so that there is no permanent addition of extra copies of DNA or extra genetic material into the cells. Let's see, I'll go back to the chat room. How long will it take the body to react to the treatment? It depends upon the condition. Let me see, I'll go up here. So I'm trying to think of one that would be relatively quick. If you were to add components of the blood system, you know, we turn over cells within our blood system very rapidly. So red blood cells and immune cells like neutrophils and macrophages turn over pretty quickly. And so that means if you added a new, let's say you were missing some of those cells. Let's say you had an immune disorder, which means you're missing some of the cells in the blood system that are responsible for immune function. If you added the blood stem cell, the hematopoietic stem cell or hematopoietic progenitor cell, it would reconstitute those cells probably pretty quickly just because the turnover is pretty fast. So in this case, it would be probably a pretty rapidly acting therapy. I'm going to be talking about dopaminergic neurons, which are the cell type lost in Parkinson's. That one would be pretty slow. You know, even after you implant these cells, it's going to take a while for them to integrate, to start reforming some of the neuronal circuitry within the brain. So this type of therapy might take months for somebody to begin to respond to it. So it's really a case-by-case basis. Okay. And I think there was one question back here on will stem cell treatment lead to a lowered cost of treatment? The hope is that for some of these conditions, they're very expensive to treat because there are no cures. And so in the case of, you know, just taking, for example, Parkinson's disease, there are therapies for it. You know, there's L-Dopa, which is a dopamine precursor that if you take it, it can begin to stimulate levels of dopamine in the brain and compensate for the fact that you're losing these dopaminergic neurons, which are the ones that produce dopamine. So you're trying to use a small molecule essentially to compensate for the loss of a natural source of a neurotransmitter. So L-Dopa is not that expensive. In some cases, however, in patients, well, in all cases, patients progressively lose their ability to benefit from L-Dopa. So you have to move on to other interventions like deep brain stimulation where electrodes are implanted. And then ultimately, you know, care for the patient suffering from this condition. So all of those, you know, do benefit the patients, but they're very expensive, especially when added over the 16 years that people live on average between being diagnosed with Parkinson's and ultimately when they pass away. So if you could replace that 16 years worth of healthcare with a single injection of a cell, that does have the potential to reduce the cost of the care, as well as significantly improve the quality of life of the patient who's received it. Okay. Another. Do you make sure you don't answer so many questions you can't get all the way through your talk? Okay, I'll jump back in. Okay, so just want to give you an example of how some of these, you know, biochemical signals can specify cell types. So this is what our nervous system for every one of us looked like, you know, many years ago. And initially there was this layer of identical tissue, and then this tissue began to pattern. And so back here at the posterior is the spinal cord. This region called the hindbrain is what gives rise to our brainstem, including, you know, the cerebellum, the medulla. The midbrain gives rise to regions like the striatum, which is what contains those dopamine neurons that are lost in Parkinson's. The forebrain gives rise to our cortex, you know, which is, you know, by and large what differentiates the human brain from lower mammals. You know, the cortex is what's responsible for a lot of our highly complex thought and cognition. So originally, all of these cells were identical. It was just a sheet of cells that made up the nervous system. And then what happened is that during development, concentration gradients of biochemical cues started getting laid down and imposed on these cells. So there's the concentration gradient of this molecule called Wnt. And if you're at low Wnt, you generate forebrain, intermediate Wnt, you generate midbrain, and high levels of Wnt, you generate hindbrain. And then back here, another gradient of a factor called FGF lays down the spinal cord. That if it's high concentrations of FGF, it ends up being the cervical and lumbar spinal cord as opposed to other regions of the spinal cord. And then you can take cross sections of these, and if you cross section the forebrain, it turns out that they're not just gradients in this axis, they're gradients in this other axis. So in this dorsal ventral, the back stomach axis, in addition to the head to toe, the rostrocaudal axis. And there are also medial lateral concentration gradients of factors. So what happens is that you end up with kind of a three-dimensional coordinate system within the developing nervous system, where a cell actually senses where it is within this Wnt gradient in this axis, where it is within the sonic hedgehog and BMP Wnt gradient in that axis, and it reads out in 3 dimensions where the cell resides. And if you have exactly the right combination of these concentrations of these cues, you can specify this blank slate early stage stem cell to differentiate into the right cell type. And just as one example, you know, for people who are suffering from a disease like Lou Gehrig's disease, or somebody who has suffered from an injury that leads to paralysis, right here is PMN, which is a motor neuron, the neuronal type that's responsible from communicating information from the spinal cord to muscle. So you could dial in the right concentration of FGF, retinoic acid, and sonic hedgehog. And if you pick the right combination, you can guide a stem cell into turning into a motor neuron, which is then a potential therapeutic candidate to treat paralyzing diseases. So that's how development works. That's how our nervous system's developed. And now if we want to do this outside the body so that we can mass produce these cells, our job is to kind of take our knowledge of development and put it into a bioreactor. And so that's where we work. And so the idea, with the cell therapy field is that you isolate or derive a cell, maybe you're making an embryonic stem cell or an induced pluripotent stem cell, and then you have to expand it, and then you have to feed it signals like these to progressively specify its ability to turn into a specific cell type. And then you've mass produced that cell and you want to implant it back into the body and engraft it to be able to treat a disease. Okay. So unfortunately, it's easier said than done. And each one of these steps or each one of these arrows just has challenges and problems associated with them. And I'll just highlight a couple here really quickly. I'm not going to read the nano font, but they have to do with the fact that these cells are very, very precisely controlled inside the body. And all you have to do is consider an experiment where, let's say you take two identical genomes, you take identical twins, and you hit the play button on their development to allow them to develop into adults. And you see that, the process is incredibly robust where the identical genome gave rise to two largely morphologically identical adult human beings. That's just remarkable that this process is so well controlled inside the body. You can turn one cell into 10 to the 14th cells in a way that is morphologically identical for n is equal to 2. It's remarkable. But once you pluck those cells out of their natural tissues, you lose all of this. And I'm showing you here information on what is known. We know that these molecules play these roles during development, but there's a lot that we don't know. There's a lot of complexity in how these various other regions or these various regions further specialize and mature and fully develop into the cell types that comprise those regions. So this is what we know, but there's a lot that we don't know. And so what this results in is that when you try to reproduce human development in a dish, you've lost a lot of control over the signals, over the information that specifies the ability of those cells to turn into the right cell types. So you have poorly controlled heterogeneous cell culture systems, complex and lengthy cell differentiation processes that can take days or weeks. And that results in, instead of having a very uniform cell type that's produced, we often end up with heterogeneous, complex mixtures of cells, which is a problem for cell therapy. If we need just a midbrain dopaminergic neuron, we don't want to implant a population of cells where only 10% of them are the right cell type, 90% of them can cause potential side effects. So a lot of what we've focused on within our group is trying to build bioreactors where we regain control over very complex processes of human development so that we can mass produce specific cell types that we would then like to implant inside the body. Okay, I'm going to skip past this and I'm going to talk about the culture system. This is now diving into our particular research. This is a culture system that we've developed within our lab over the past 10 years or so. And this is kind of a meld between chemical engineering and biology and stem cell biology. So the goal is that, human development is obviously occurs within 3 dimensions. We are 3-dimensional organisms, but a lot of the cell culture systems that people use involve adhering cells onto 2-dimensional rock hard plastic. And I can go into the reasons for that. They're largely historical. But as a result of taking the cells out of this comfortable 3-dimensional pillow of a tissue and putting it on rock hard plastic, a lot of control is lost over the ability to control the expansion and the differentiation of those cells. So what we've worked on is trying to develop scalable systems that you can put into a bioreactor where you can trick a cell into still thinking it's inside of its nice, comfortable 3-dimensional tissue. So a lot of our body, it turns out, is water, right? We're on the order of 70 to 80% water. So a lot of the tissues inside of our body are what are called hydrogels, soft gels, where the majority of these tissues is water. Our knees are really, really good examples where our knees are essentially collagen gels, a protein gel made of collagen, and that serves as a great cushion so that it can absorb a lot of load. Or the lens of the eye is another good example. It's a hydrogel that's mainly water, and that's important for the ability of light to penetrate through. So there are lots of hydrogels inside the body. A lot of the tissue inside the body is a hydrogel. So we began to develop a synthetic hydrogel, a polymeric hydrogel for embedding cells within it. And this hydrogel has an interesting property that it's a liquid at lower temperatures. So you can see this thing flows really easily. So at low temperatures, like on ice, we can take individual cells and just kind of mix it together with this liquid. And then once you elevate the temperature above 22 degrees or all the way up to 37 degrees, which is the temperature of our body, of course, then this turns into a three-dimensional gel where cells are now suspended inside that gel. And what that enables us to do is to kind of encapsulate cells inside of this environment, inside of this niche, this microenvironment, if you will, at 4 degrees, elevate the temperature so the cells are captured inside the gel. We can expand them then inside of this nice, comfortable pillow of a hydrogel. And then when it comes time for us to recover the cells, all we have to do is stick them back on ice. The gel, the polymer turns back into a liquid. We can harvest the cells, we can spin them out. And then if we want to, we can either break up the cells back into individual cells and then put them through this loop many times so that you can get exponential expansion of the cells, or maybe you harvest the cells when you're done and then start using for therapeutic application. So this has a large number of advantageous properties, including the fact that, chemical engineers would call this scaling up into three-dimensional culture as opposed to scaling out two-dimensional culture. And it's highly preferable to do a scale up rather than a scale out if you're trying to increase the scale of the culture. And I just want to give you a sense of the challenge associated with scaling up culture of cells. And that's the fact that if you just take Parkinson's disease, I'm going to keep coming back to that as an example. There are around 1 million patients in the US who have Parkinson's. They will require on the order of several million cells each. And if you do the math, you'd be dealing with on the order of dozens of football fields worth of two-dimensional surface area to grow the dopaminergic neurons for implanting into people. Whereas if you move up into a third dimension, by going into a hydrogel, for example, it dramatically decreases the size of a bioreactor you would need to be able to treat this condition. So we started synthesizing these materials. We started growing cells within them. And we began to find that we got very, very robust growth. And you can see from day 1, we have single cells, and by day 6, we have very large clusters. So there's a very rapid expansion of these cells. And I'll mention as well that the conditions are what you would call completely chemically defined. It's a synthetic polymer, a cell, and a culture medium, a broth of nutrients that is fully defined. And for people who want to begin to scale up production of a product like this for clinical application, having fully defined scalable conditions is going to be really important as you begin to head into human clinical trials. So this is what it looks like in 3D. We can embed cells inside this material, extrude it, for example, from a syringe into a warm broth of fluid so it gels. And if you zoom in on the spaghetti, you can see that over time you have individual cells, and then these gradually grow into large clusters of cells inside this three-dimensional snake of a spaghetti embedded inside of a bioreactor. The cells divide very, very reproducibly. They divide 20-fold every 5 days. And we started doing this many, many times in a row to kind of test the ability of the system to expand the cells reproducibly. So every single one of these, what are called passage numbers, corresponds to 5 days of expansion. And each time we got a 20-fold expansion. So if you take 20-fold and raise it to the 55 power, it turns out that that's a 10 to the 72nd fold expansion of these cells over 280 days. So that really massively addresses the scalability problem. We're not going to have any problem generating enough cells to be able to treat whatever condition that may be of interest to us. And I'll mention as well that the cells are very stable throughout that expansion process. They stay stem cells. They maintain, after you expand them for, 280 days inside this material, you then have the ability to differentiate them into many cell types in the adult body. So they maintain this capacity for pluripotency. They can turn into, in principle, every cell type within the adult body. And furthermore, you want to make sure that they're genetically stable, that they're not acquiring mutations over time. And we find that these cells, this is a layout of their chromosomes, their chromosome stability is quite high. So you can expand them indefinitely inside this gel, and their chromosomes, their genome is stable. Okay, really quick question. Does oxygen transport by diffusion in the fixed gel matrix limit the maximum metabolic rate? So Brian's bringing up the issue that oxygen has a very low solubility in liquid. So it's hard to transport oxygen long distances inside of a liquid. And that's why our blood system is so specialized as a piping to be able to transmit oxygen from our lungs to the tissues throughout our body. It's to overcome this low solubility limit. We need a plumbing system to rapidly transport oxygen. Within this gel, show you an example here. So in the dark regions, this is liquid. So we do have the ability to convect liquid, to mix liquid, and you could even sparge oxygen bubbles through it. That's not a problem at all. So you can maintain the black regions at as high an oxygen concentration as possible, convectively. And then oxygen still has to diffuse from the surface of the gel to the interior. And that diffusivity, because this is a hydrogel that's over 90% water, it's the same as the diffusivity of oxygen inside of water. So it's relatively high. And what we can say is that we find that cell division rates in the middle of these fibers are identical to that near the outside. And as I'll talk about in a second, is we can also differentiate the cells inside these fibers. And we find no gradients of differentiation across the dimensions of the fiber, which to us at least indicates that we're not seeing significant nutrient transport limitations or oxygen transport limitations in the system. Okay, oops. Okay, so this indicates that we can generate very large numbers of cells, but these cells are not yet therapeutic, and we need to turn them into the cell type that potentially could be used for therapeutic application. And so I keep coming back to Parkinson's disease because we work on it. And I want to give you the state of the art in Parkinson's cell therapy around 15 to 20 years ago, and then talk about how the field can do better. So Parkinson's disease is due to largely the loss of a population of dopaminergic neurons in the midbrain, a region called the substantia nigra. And the idea came up, well, these cells are being lost. Can you implant midbrain dopamine neuron replacements for them? So you can try to rebuild that neurotransmitter signaling system in the brains of Parkinson's patients. So back, 20 plus years ago, 20 to 30 years ago, investigators began to explore this idea, and the only source of dopaminergic tissue that they could get a hold of at the time was from aborted fetuses, from abortion. So what they would do is that they would accumulate this tissue, and in some cases it took up to 10 fetuses worth of tissue per adult Parkinson's patient, and they would cut out the dopaminergic region, mix it together, inject it into the brain, and try to repopulate this population of cells. And they found both good news and bad news. And so the good news was that in some situations, in some cases, the patients experienced absolutely clear, pretty remarkable recovery. And that recovery was long-lived. It really began to deliver on the potential of a one-and-done cell therapy that a single administration could result in a long-term benefit. The bad news was that this is really it's an unsustainable, untenable source of cells. I don't even want to get into kind of the ethical, awkward discussion about how you would source these cells. And in addition, it's a highly heterogeneous source of cells that if you have to surgically isolate from 10 fetuses per patient, this is not going to happen the same way twice ever. And as a result, there was a lot of heterogeneity, a lot of variability in the outcomes. And in some situations where the graft stayed at the site of injection, the patients didn't benefit at all. In some situations where this surgical isolation of the dopaminergic region unfortunately carried along with it some contaminating other populations of neurons from the surrounding regions, this actually led to neurons in the adult brain in places where they had no business being, at the injection site, and this actually led to side effects within patients. So the good news is that in principle, a dopaminergic cell replacement has strong potential, but the bad news is that this is a very poor source of cells to do that. But now, if you fast forward to the current time, we now have plentiful, in theory, infinitely renewable sources of stem cells that can be coaxed into differentiating into a midbrain dopaminergic neuron. So based upon, I'm going to zoom back really quick, based upon what people have learned from human development, well, the midbrain is specified by this combination of signals and cues. So investigators began to, in cell culture, try to imitate those recipes, those combinations of cues. And those are shown right down here. In order to coax over a period of around 20 days, 25 days, a population of pluripotent stem cells into differentiating into midbrain dopaminergic precursors. So this was done originally in 2 dimensions, and it involved a lot of messy stuff, a lot of poorly defined cues, animal and human derived proteins. So we took what had been known in the literature and we scaled it up into a 3D gel and did it under completely chemically defined conditions. So it could be done reproducibly today, tomorrow, next month, next year, the same way. In other words, that gel that I showed you a little bit earlier can serve as a platform both for expanding the cells, but if you then change the broth, you change the nutrients, you change the signals in the cell culture liquid that you add, you can then convince the cells to turn from a pluripotent stem cell into progressively being specified into a midbrain dopaminergic neuron. So this is just some data showing that we were able to do that, to guide these cells into showing the markers indicating that they are midbrain dopamine neurons. The yield is extremely high. So the number of cells we end up with per milliliter of hydrogel is extremely high compared to the volumes you would need of culture or of a bioreactor in a 2-dimensional cell culture system. And it turns out that our bodies are 3-dimensional, they're not 2-dimensional. And so if you do this inside of a 3-dimensional soft gel, you actually get better differentiation. A higher number of cells differentiate into the right lineage, into the midbrain dopaminergic fate, compared to a number of other alternative contaminating cell fates. So you generate a purer product. And finally, just to show the idea that once you do this in 3D, that the cells have the potential to differentiate all the way into mature dopamine neurons. We took this cluster of cells that we'd grown in 3D and put it back on 2D, and you can see that very rapidly it extends the wiring that would be a hallmark property of a mature neuron. And then we can kind of characterize these cells and show they have the right electrophysiological signaling properties. So we collaborated with somebody in the College of Chemistry, Evan Miller, and what Evan does is that he develops what are called voltage sensitive dyes. And so we know that neurons fire action potentials. They fire electrical signals along these fibers. And as they do that, the voltage across the membrane is going to change. And so he developed a dye, a chemical fluorescent dye that changes its fluorescence properties as a function of voltage. So you can actually watch movies of these wires firing. And you can convert these movies into something that looks like an electrophysiological signal. And we find out that in 3D, we end up with 5 times as many cells firing in a pattern that would indicate that they're actually dopamine neurons. So we not only can generate cells in much higher yield and purity, but also more functionally valuable, having the function of a mature midbrain dopamine neuron. And then we began to take these and implant them into the brains of animals. And I'm going to skip ahead really quick because I think we're running out of time, and I'm going to jump to the punchline. And what we did was that we began to implant these into the brains of animals that were models of Parkinson's disease. So just to remind you, Parkinson's is due to the loss of this population of midbrain dopamine neurons. If you introduce this small molecule toxin, it's called 6-hydroxydopamine, it specifically kills off that population. And now you've created an animal model of Parkinson's. The animals have motor symptoms that are reminiscent of Parkinson's patients. And one of these is called a forelimb akinesia. It's just a motor symptom that indicates whether these animals are behaving like a Parkinsonian animal. And in this score, this motor score, lower is worse, higher is better. And so 2.5, if you could draw a dotted line here, is an animal that was never receiving this lesion. So a completely normal animal would be around 2.5. So if we do just a control sham injection down here, you can see it's very poor. If we go all the way up and implant three cells that were manufactured in our 3-dimensional system, along with a couple of other helping cues, which I won't go into detail unless we have time in Q&A, you can see that all of a sudden we're actually completely repairing this region of the brain to the point where it's statistically indistinguishable from animals that were never injured. So I'll close out by saying that we've done this not only for Parkinson's disease, we've done it actually for oligodendrocyte precursor cells, which are the cells that are lost in multiple sclerosis. We've done it for what are called striatal neurons, which is the cell type that's lost in Huntington's disease. And based on these advances, last December, we started a company called Accent Biosciences, where we're beginning to develop this technology to start going into human clinical trials, which we look forward to doing so within the next 2 years. So just to summarize, we develop platforms for being able to manufacture stem cells and manufacture therapeutically relevant cell types from stem cells, where we can create highly controlled, reproducible, scalable, cheap, safe bioreactor platforms where we can expand these cells indefinitely and differentiate them almost into limitless quantities of cells that can then go into human beings. And we anticipate that Parkinson's disease will be our first target as we move this towards human clinical trials. So I'd like to acknowledge the people who actually did the work. This is my current lab, but the people who did the work that I showed you today, it was the product of Amruf, Badri, Barbara, Leo, Gonzalo, Tandis, and in some cases collaborating with Sanjay Kumar in bioengineering and Evan Miller in chemistry. So thanks so much for your attention. I don't know if we have any more time, but if we do, I'd be happy to answer any other questions.
David, let me begin by thanking you. It was fascinating and probably a very relevant topic for some of us of this age. And let me start by asking a question.
Mm-hmm.
Take your example of Parkinson's, which is caused by the fact that normal cells are dying off in the brain.
Mm-hmm.
So you add new ones. What's going to keep them from dying off if the brain can't sustain its own original one?
Yeah, that's a great question. And people have seen that actually. So in those old clinical trials where they used fetal dopaminergic tissue, those were 20, 30 years old. And so some of those patients have passed away, and they donated their tissue to the scientists so that they could actually study the cells that were implanted. And they found in many cases the cells persisted for over a decade. That's the great idea of one and done, single administration. And because DNA is stable or cells are stable, they can persist for long periods of time. And they found that some of those cells were looking like they were starting to get some of the symptoms of Parkinson's. They had so-called Lewy bodies inside of them, but it was slow. If you think about it, it takes somebody 50 to 60 years to develop Parkinson's the first time. And if you implant brand new cells, they might slowly get sick as well. But if you can buy somebody 10 or 15 years of delaying the onset of Parkinsonian symptoms, that's 10 or 15 years of high quality of life before, perhaps eventually those cells start getting sick just like the original cells did. And by that time, maybe, if I have Parkinson's, my heart gives out on me anyways. But I get to enjoy 15 years without Parkinson's.
Excellent. Sort of optimistic view of what will happen. A couple more related questions have come in. Once you have your cells that you've produced in your boilers, what are the challenges of actually injecting them, presumably into the right place, the right amount?
Yeah, we need delivery systems as well. And I mentioned to you that one of the standards of care for Parkinson's is so-called deep brain stimulation, where you actually implant electrodes in order to kind of deaden some regions of the brain that become overexcited in Parkinson's disease. So what that means is that people who have received a deep brain stimulation are walking around with electrodes inside their craniums for long periods of time. So being able to implant something that looks like a needle is relatively easy for a neurosurgeon. So it would be possible, and it has been shown that it is possible to use a needle system to disperse these cells into the regions they need to be. And that's a relatively short surgery, and it's a relatively straightforward one for a neurosurgeon.
And there's no risk that they'll escape and do damage elsewhere?
The cell types, that's the ones that we're implanting no longer divide. And if they ended up escaping the brain, they'd probably just die.
there's a question relevant from Martin Babrowski. It's been recognized there's a long prodromal phase of Parkinson's. Would you theorize that treatment should be early or when more typical symptoms develop?
Yeah, it would be, that's a debate within the field because, the FDA's view, and I'm not criticizing them, I think it's reasonable, is that you want to do no harm to a patient, right? And so typically the initial clinical trials would likely be, you'd be working with later stage patients who, have progressed to the point where they don't have as much to lose. Because you wouldn't want to take an earlier stage, still pretty healthy patient and expose them to risk of a new therapy. Because, I don't think anything's going to happen, but you never know. And so you want to make sure that you do this in as safe a way as possible. So likely initially it would be used to treat later stage patients. And then once strong safety had been established, then you could begin starting to treat earlier stage patients where they have more and more to gain.
Related, how do neurons injected without organization form the proper connections for function?
Mm-hmm. Yeah, so they're injected, the cells would be injected locally in a region where their axons. So, shown here in this image, these are midbrain dopamine neurons. They're extending these processes, this wiring. So ordinarily, these axons will project to and form synapses with cells in the striatum. And then once the dopamine neurons die, those axons die, and then the striatal cells lose that input, that dopaminergic input. So you would put the cells back into the striatum, and they'd re-extend these axons, your human embryonic stem cell derived dopamine neurons. And as long as you do that in a way where the axons are pretty well spread within the region where they've been depleted, then it appears you can get pretty ameliorative, pretty beneficial outcomes to the patient. So it's not as important to get the exact structure. You just need to make sure that the cells spread out from the injection site pretty well.
Martin follows up with, and I'm not sure this is the purview of QB3 or not. Care to comment about objections to stem cell therapy based on philosophical, theological, and societal concerns about eugenics and treatment that would be for cosmetic reasons?
Yeah, I'm not crazy about cosmetic applications of cell therapy. I think that it's probably up to a philosopher to decide exactly what the word illness means. But I think that the society has a pretty good sense of what an illness is relative to a healthy individual.
Well, certainly when we talk about Parkinson's, this is way beyond cosmetic.
I wouldn't feel, I don't think that using it for, cosmetic surgery on the face is something that would really excite me as an individual, because I don't find that as much of an unmet medical need compared to people who are suffering from a degenerative disorder like congestive heart failure.
Yes.
So I think that most people who are developing therapies involving stem cells are probably like-minded to me, but there are, you have heard of predatory stem cell clinics that are out there that are doing it for the wrong reasons.
Yes. You haven't mentioned cancer, and there was a question of, do these procedure methods have any applicability to treating cancers?
Yes. So one of the most exciting developments in the cancer field over the past 10 years, well, really over the past 20, 30 years, has been cancer immunotherapy, where you can intervene to teach your immune system to recognize that a cancer is foreign. Cancers are systems that have, unfortunately, very ingenious ways of blunting an immune response and shielding themselves from the immune system. And so if you can just get the immune system to recognize that the tumor is foreign, you can then really wire up the immune system, which is quite powerful, to attacking and eliminating the tumor. So one way to do that is to add a T cell. So a T cell is a cell type that's really responsible for a lot of the effector, a lot of the attacking function of the immune system. So it is possible to engineer a T cell to recognize a tumor, to recognize a cancer. And these are so-called CAR Ts. There are 2 FDA-approved CAR T therapies in the United States right now. So people are trying to turn pluripotent stem cells into T cells so they can use them for cancer immunotherapies as T cells that attack tumors.
which presumably is important for those of us who are in the 65 and above, and therefore more at risk for the coronavirus because of our reduced immune responsiveness.
Yeah, there has been talk as well about engineering the immune system to recognize signals from the coronavirus, so that, you know, it takes our adaptive immune system. You know, when you get a vaccine, it takes, you know, the vaccine, or it takes your immune system, you know, a couple of 2, 3, 4 weeks to ramp up to the point where it could benefit from, let's say, the flu vaccine. So even after you've gotten the flu vaccine, it takes 2 or 3 weeks for your immune system to really learn how to attack the flu before it's fully strengthened. And unfortunately, within that 2 to 3 weeks, a lot of bad stuff can happen with SARS coronavirus too. So if you could actually teach the immune system and gear it up to specifically attack SARS coronavirus 2 quicker, then that has a lot of benefit. So people are talking about immunotherapy treatments for SARS.
Obviously, we should hurry up.
Yes.
Donald Mastronardi points out quite appropriately that the stem cell initiative is up for renewal. What role is QB3 playing in getting this whole program renewed?
Yeah, that really falls under the responsibilities of the Stem Cell Center, which I can comment on because I think I'm still technically director of the Stem Cell Center. I've stepped up to QB3.
Lucky you.
And the next director of the Stem Cell Center has not yet been identified. So over the past several years, I've been serving on the scientific advisory board to Bob Klein, who's the person who is the architect of Proposition 71 and is the architect of this renewal proposition that's going to be on the November 2020 ballot. So, you know, they have been very active over the past 2 to 3 years, getting the signatures, you know, 800,000 signatures to get it onto the ballot, and are now, you know, really proposing it and trying to get the word out that this is something that voters should vote for. You know, obviously, as somebody heavily involved in stem cells, this is something that I'm going to vote for. But everybody, of course, needs to make their own decisions based on the facts. So at the very least, I hope that the Stem Cell Center can try to educate people so they can make their own decisions this November.
Well, thank you. And perhaps that's an ideal way to end with the hope that everyone who listened to this lecture will not only go out and vote in favor of the stem cell initiative, but talk to their community and get more people involved. It sounds like a very promising future for all of us.
Paul, this is Carrie. Before you do close off, I'd like to see if we can unshare the speaker slides and see if there's anybody that physically needs to raise their hand and ask a question that may not be using the chat feature. And make sure that we did get all the questions answered in the chat box. We can unmute you if you physically raise your hand. Or we can, for those people without their video, we can unmute all and see if anybody has a question.
Al is special.
Okay, I'm trying to unmute Al. Just give me a second here. It's not cooperating.
Can't he unmute himself?
Can you try? It won't let me do it for some reason. I've unmuted you twice.
Try to unmute yourself, Al. There you go.
What do you think?
How's that?
Perfect.
There. Okay, go ahead.
It is not clear to me whether all of the pluripotent cells need to be fertilized, or whether some of the initial cells that you were talking about are unfertilized.
They would have been fertilized previously in their history. So they went from
all of them are.
Yeah, from a zygote to an embryo, and then the pluripotent stem cells were isolated from that embryo. So they were historically fertilized, you know, back, you know, a number of weeks earlier, or actually days earlier in human development, prior to isolating the embryonic stem cells.